Bismillah ar-Rahman ar-Rahim. This is the presentation made by Professor Dr. Iman Sadi, Professor of Histology, Faculty of Medicine. Now we will describe the practical class covering the peripheral nervous system. The peripheral nervous system is formed of nerve trunk, ganglia, as well as nerve endings. This presentation is only the nerve trunk and ganglia, while the nerve endings will be in another separate video. Before you start to see this practical revision, you should revise the theoretical part involving the nerve trunk as well as the nerve ganglia, and the link for this lecture, which will be lecture 2, will be present in the description, inshallah. Now this is the first slide. It shows the section in the nerve trunk stained by hematoxin and UC. As you see, we have three components for the nerve trunk. First, we have this connective tissue covering the whole nerve trunk, which is called epineurium, and it is formed of dense connective tissue. Then we have the nerve fibers surrounded by, in the form bundles, surrounded by the this connective tissue called perineurium, and this is formed of tightly joined cells. Then in the center of the bundles, in between the nerve fibers, we have a very pale and connective tissue called endoneurium, formed of reticular fibers. In this picture you can see the epineurium, and these are blood vessels. We have the perineurium surrounding the bundles of the nerve fibers, and here inside we have endoneurium. This is a higher magnification for the previous slide. On the right side you can see this is the connective tissue formed of the tightly joined cells here, which is the perineurium, forming a type of a barrier, blood-nerve barrier. And here inside the nerve bundle we have the nerve fibers formed of axon surrounded by an area for myelin sheaths, and then we have the neurolemmal sheaths, and you can see the nuclei of the Schwann cell. In between them we have a very delicate connective tissue, so it is very pale, formed of reticular fibers, and it is called endoneurium. In this we have these labels. Number 1, this is the nucleus of the Schwann cell surrounding the nerve fiber, and it is only one in the section. And we have number 2 will be the nerve fibers, and number 3 will be the axon or the axis cylinder in the center of the nerve fiber. Of course you cannot see the myelin sheath because it is dissolved in the alcohol during preparation of this section stained by hematoxylin and AUC. And in this picture also you can see these are the nuclei of the Schwann cell, and this is the axon or axis cylinder, this dark spot inside, and this is also the same dark spot, so the same axon. Of course, in the diabetes mellitus, the demyelination of the nerve fibers, peripheral nerve fibers, will lead to impairment of nerve function seen in the advanced cases of diabetes mellitus. This is another picture for the slide stained by osmic acid. The osmic acid is a stain that dissolves in the myelin sheath only and gives it a black color. So you can see here the bundles of the nerve fibers, and this is a lightly stained tissue. It is the tissue covering the nerve fiber, which is called the epineurium, and this tissue surrounding the bundles is called the perineurium, and these are the bundles, and inside we have the nerve fibers, and in between these areas which are white in color, they are representing the endoneurium, while these dark circles, they are representing the myelin sheath of the nerve fiber. If you look at the high power, you can notice the black rings representing the stained myelin sheath by the osmic acid. The osmic acid dissolves in the myelin sheath and gives it the black coloration. And in the center we have this empty area representing the site of the axon or axis cylinder, Of course, we have this white area or lightly stained area. They are stained non-specifically, just a very faint stain. This is the endoneurium, and also non-specific stain for the perineurium in this picture. And this is one bundle only of the nerve fibers. Let's move to the other subject, which is the ganglia. The ganglia, we have two types of the ganglia. We have spinal and cranial. This is one category, and we have autonomic. This is another category. The autonomic is divided into sympathetic and spinal, and they are similar in histology to each other, while the spinal and cranial have a different structure. Here, this is the spinal ganglia. How in the practical exam you can diagnose the spinal ganglia from the sympathetic ganglia? In the theory, we have 10 differences, but in the practical, we have just very evident 3 differences. These differences are actually SAS. Which is SAS? SAS is the shape of the cells, the arrangement of the cells, and the size of the cells, or size, arrangement, and shape, SAS. What about the size of the cells? As you see here, the cells here are of different sizes. We have small size, we have moderate size, we have large size, so we have different sizes. Also, as a general look, the cells are more or less or relatively in comparison with the sympathetic, they are large in size. So for the size, we have 2 points. Then arrangement. The arrangement will be in groups. This is group, and rows. This is a row, and this is a row. Then the third point, it is the shape. The shape of the cells, these cells are classified as pseudo-unipolar. They have this globular structure for the pericardium or for the body, and they don't have any projections or dendrites, so they are pseudo-unipolar. This is the 3 rapid points for the rapid diagnosis in the practice. Also, we have a very important point to remember in the exam, the fibers. The fibers here are actually myelinated nerve fibers. So these fibers are myelinated nerve fibers, and this is a major difference between the spinal ganglia and the sympathetic ganglia. So we have here the myelinated nerve fibers in between the nerve cells. SAS and myelinated nerve fibers. Here another picture for the spinal ganglion, and in this picture you can notice another additional structure covering the ganglia, which is the capsule. It is present in both spinal and sympathetic, but it is thicker in the spinal than in the sympathetic. Then we have here the nerve cells. Don't forget SAS. We have, as regards the size, we have different sizes, and more or less they are of larger size. As regards arrangement, they are arranged in groups. This is a group and this is a row. As regards the shape, they are pseudo-unipolar. This is the globular structure of the nerve cell, no dendrites, and this is the appearance of the myelinated nerve fibers passing in between these groups and rows. Now try to label the different structures. Starting from the center, we have this is the nucleus. This is the nucleus. Of course, this is in the center of the nucleus is the nucleolus, and this is the cytoplasm here, full of granules. Here we have granules, which are nestle granules, and then we have the surrounding cells, which are called satellite cells. Don't forget satellite cells. These satellite cells, they are surrounding the nerve cells in the ganglia, and they are belonging to the neuroglial cells. Here we have, as we see, this is the nucleus, and these are the satellite cells belonging to the neuroglia. And don't forget that the nerve fibers here are with myelin. They are myelinated nerve fibers. Another picture for the spinal ganglia stained by hematoxin and leucine. You can see here these important structures. We have the thick capsule, we have the cells. Don't forget size. What is size? It is the size of the cells, arrangement of the cells, and the shape of the cells. In between them, we have the myelinated nerve fibers. This is a picture for the spinal ganglia, but in this slide it is stained by silver. The silver will stain the Golgi apparatus and the neurofibrils. So we have these brown bodies representing the pericardium rich in Golgi apparatus, and also we can see here some of the fibers containing the nerve fibers containing the neurofibers stained by silver. The silver will give the brown coloration. Now, how can you diagnose this is the spinal? By following the rule of SAS. We have as regard the shape and the arrangement and the size of the cells. First of all, the arrangement is very evident, which is in the form of rows and groups or collections. So this is a very easy way to differentiate from the first look between the spinal and the sympathetic. Of course, we have on the surface the capsule, but it is stained non-specifically, and the nerve cells will be stained by the brown due to the presence of well-developed Golgi apparatus. In between, you can see these brown lines representing the axis cylinder, which is rich in neurofibrils, and these neurofibrils are stained by the silver. So silver here will stain these two structures, Golgi and neurofibrils, by the brown coloration. Here, this is a very good and magnified picture for the spinal ganglia stained by silver. You can see here the bodies stained by the brown color, by the silver stain, due to the presence of the Golgi. Of course, we have also neurofibrils inside, but here the Golgi is very prominent cause for the brown coloration of the pericardium. Also, we can see these neurofibrils present in the nerve fibers. These are nerve fibers cut transversely, these are cut longitudinally. So we have these dark brown lines representing the axons, which are rich in neurofibrils that will take the brown coloration of the silver. So this is the capsule. These are the cells arranged in the form of groups and rows, and these are the nerve fibers. Don't forget, they are myelinated nerve fibers. Here, this is the picture for the sympathetic, but this slide is stained by hematoxylin and UC. So you can see here the capsule. The capsule here is thin compared to the thick capsule in the spinal ganglia. Here we have SAS, which is the size of the cells. They are almost similar, and they are relatively smaller compared to the spinal ganglia cells. We have A, which is the arrangement, which is the scattering appearance of these cells, and we have the last S, which is the shape. Here the shapes, they are not globular or rounded, they have pointed ends due to the emerging dendrites from this multipolar nerve cell. Here we have sharp or acute angles due to the presence of the dendrites coming out from this pericardium, as these nerve cells are classified as multipolar stellate. Here in between, you can see these areas representing the site of the nerve fibers. The nerve fibers here are unmyelinated. Don't forget, the sympathetic ganglia is characterized by having unmyelinated nerve fibers, in contrary to the spinal, which is provided with myelinated nerve fibers. Here we have also the neuroglial cells surrounding the nerve cells, which are called satellite cells. Don't don't make a mistake between stellate cells and satellite cells. Stellate means they have star shape, and satellites they have the appearance of a circle surrounding the body, like the satellite in the outer space. Here we have the magnification of this section. We have the bodies of the nerve cells. They have the nucleus, they have the cytoplasm, and the shape of the cells here is not globular. It has these corners. Look at these corners, 1, 2, 3. They are not smooth or rounded. Here it is the multipolar stellate cells. Stellate looking like a star. But the cells in between is called satellite. Satellite cells. These cells, they are surrounding the nerve cell like a satellite. They are neuroglial cells. يبقى هنا دايما في ميستيك ما بين stellate زي النجمة، satellite اللي هي عاملة زي الايه الساتلايت او القمر الصناعي اللي حوالين هذه السيلز. In between we have what? We have unmyelinated nerve fibers, because the nerve fibers here are unmyelinated but provided with neurolemma. Here we have the sympathetic, of course, stained by silver. Why? Because it is brown in color. Why it is sympathetic? By the scattered arrangement. Scattered arrangement, and after that we have S, which is the size small, uniform, and the shape. Here the shape is not evident. يعني لازم نشوفها higher magnification عشان نشوف ان الشيب هنا is stellate shape. And this is of course the capsule. Capsule is thinner compared to that of the spinal ganglion. Now we have this magnification, and you can see here the SAS very clearly. Here we have the size of the cells. They are almost all similar, uniform, and they are small. The arrangement is scattered, and the last S is the shape. They have here the sharp corners. They are not globular. They are not rounded like that of the pseudo-unipolar cells of the spinal ganglion. After that, if you have a label on these fibers, you should mention that they are unmyelinated nerve fibers. Here we have the axons here. They are very close to each other. You don't have myelin here. Don't forget, sympathetic ganglion بتسرق المايلين. Here we have also a section stained by silver. Why? Because it is brown. Now we have these nerve cells, and these nerve cells appear to be scattered. Not only that, but we have some angulation, some sharp corners, meaning that they are what? Stellate in shape, multipolar stellate. Here we have this label. When we have this label on a structure stained brown inside the body of the nerve cell, so you should mention that it is the Golgi apparatus here. And this is the nucleus. In between these nerve cells, yes, اهو يبقى ده هنا unmyelinated nerve fibers. Here are present in between the bodies of the nerve cells. And you have a scattered appearance, so this is a sympathetic ganglion stained by silver. Thank you very much. And now don't forget to see the video for the trial test of this nervous tissue. And if you are not subscribed to my channel, please subscribe and activate the notification bell to get notified when I post a new video. Especially in the coming days, I'm going to post, inshallah, 3 videos a week to help you in the practical exam. Don't forget to like the video. Thank you very much.